In the second part of chapter 11, we're going to be looking at the forms of reproduction and the role that meiosis needs to play. Now, recall that there are two basic forms of reproduction seen in living things. Asexual forms of reproduction, in which we see no fusion of gametes to form a new organism, and sexual reproduction that involves the fusion of gametes. Asexual reproduction is done by single-celled organisms, and basically, as we talked about in the last chapter, in chapter 10, they reproduce simply by mitotic division. However, it's not just single-celled organisms that can reproduce in an asexual process. Multicellular organisms can also reproduce asexually, such as budding by a hydra. We see here a hydra that we'll look at in the second half of this course next semester, and we can see that by new cells growing by mitosis, a bud can be formed, and this bud represents cells that are essentially clones to the parent, and eventually this bud can break free from the parent, and we now have had an asexual reproduction. Now, these organisms that are produced asexually are clones. They're genetically identical to the parent. And if you think back to our talks about evolution, we need changes. Genetic variation is necessary to allow the survival of life on Earth. So how do these changes occur in organisms that reproduce asexually? Well, through the processes of mutations, changes in DNA sequences that happen by accident and so forth, as the DNA is replicating itself, etc., can all cause changes called mutations that can promote some genetic diversity. Now, for the context of the rest of this chapter, we're going to be referring and looking at sexual reproduction. Sexual reproduction, the process of sex, comes from the Latin word sexus, which is interesting in that the word sexus literally means to divide or separate, as in separating the different sexes, male and female. So in sexual reproduction, we're going to see the fusion of gametes. And of course, in animals, male gametes are called sperm, and the female gametes are known as the ova or the egg. These sperm and eggs will fuse together during the process of fertilization to form this zygote. And if you look at the ploidy levels of these, the sperm and the egg are both haploid. And when fertilization occurs, they literally become one cell, and we see a parental homologue from the sperm and the egg contributing to form this diploid zygote. Now, parents give rise to offspring that have unique combinations of genes, especially organisms that are produced in this sexual fashion. These are not clones. So two essential questions have to be asked. Well, how did this genetic variation arise, and how are offspring produced with the correct number of chromosomes? If we're going to fuse two cells into one, you would think that the genetic material would be doubled. And in fact, it is. But how do we go from diploid back to haploid? Now, this is very important to understand, because if you think about humans, our normal somatic body cells have 46 chromosomes. This is because these chromosomes occur as homologous chromosome pairs, one maternal chromosome and one paternal chromosome. And each homologous chromosome will carry genes that control the same inherited traits. And as we'll see in a few later chapters, we'll look at how these genes are expressed depending upon the traits that each homologous chromosome pair contributes. Cells that have pairs of homologous chromosomes are said to be diploid, like our somatic cells, and cells that have half of a homologous chromosome pair are said to be haploid. In humans, these are the gametes, sperm and egg. Now, you may have heard of these things. These are karyotypes. Karyotypes are used to observe cells' ploidy levels. These are ordered displays of condensed chromosomes, and what they do is they arrest the cells during metaphase. And if you remember metaphase, where the homologous chromosome pairs are aligned and attached to the mitotic spindle, they are at their most easily separated at this point. And so the karyotypes are going to be used then to screen for genetic anomalies, such as Down syndrome. Karyotypes can also be used to determine gender, sex, male or female. And this has to do with, in humans, like many animals, there is a pair of homologous chromosomes called the sex chromosomes. In humans, it's the 23rd pair. In other organisms, it may be a different pair. Now, the sex chromosomes are separate from the other chromosomes, and the other chromosomes are known as autosomes. And so in mammals, if the sex chromosome pair contains an X and a Y, then the gender is male. If the sex chromosome pair contains two X's, then the gender is female. So here's a look at a karyotype of a human male, and we can see then the pairs of homologous chromosomes. And if we look at the 23rd pair in the corner here, we see one X and one Y. I know you don't recognize this as a Y shape, so to speak. It just has to do with the position of the centromere very near one of the ends. The centromere is not always in the direct center of the chromosome. If we look at a human female karyotype, we don't see a Y chromosome, we see two X's. And so what this really translates to then, if you think about it, and we'll talk about this more in the next couple of chapters, in mammals, the male gametes are either going to carry an X or a Y. And when the male gamete contributes this X or Y chromosome to the zygote, it will actually determine the zygote's gender.